Hey, hi everyone. Good morning. I think it's nice that a couple of you say good morning in the chat. I just got my third Pfizer booster shot. Well, no, my third shot, which is my booster shot yesterday, and I'm feeling really achy, right? So fortunately, not too bad. I don't think I have a fever or anything, but I definitely feel kind of achy and slower, which is kind of a bummer because we have a lot of cool stuff to talk about today, right? So I'm going to try to sort of stay sharp and get us through it. All right. So last couple lectures, we've been talking about how cells take glucose as the raw fuel molecule, right? And how through the process of glycolysis, through the three stages of glycolysis, the cell is able to get two molecules of ATP out of that glucose. And we said, why is that important? Why do we have to start with glucose and then make ATP from it? Well, glucose is the raw fuel source that is put together by photosynthetic organisms on this planet, things like plants, for example, that take the abundant carbon dioxide, the abundant water, and the energy of the sun, and rearranges those bonds of the CO2s and H2Os to make glucose, right? And therefore, the cell then needs to take that glucose and convert it into ATP, because the ATP is the actual form of that energy in the form of that third phosphate bond on the ATP, right, that all of the various proteins like enzymes inside of the cell is geared up to use. Right? All those examples we've shown of how certain enzymes that act as ATPases bind to ATP, break that third phosphate bond off, and that breaking of that bond, right, the liberation of those electrons that were in a higher energy bond there, right, ends up powering different things, right, like the movement or the changes in shape of different proteins that allow them to carry out certain jobs, like the active transporters that we learned about when we were studying membrane proteins. And we said how important those are because those are able to pump things, pump solutes against a gradient, oftentimes so that gradient can be taken advantage of later, allowing things to flow back downhill through the membrane and using that as a source of power to then also accomplish other types of molecular movement. So we said that all life on Earth began in anaerobic conditions, right? There wasn't much O2 in the atmosphere at all. So therefore, in order to get ATPs out of glucose, early life had to do it without the presence of oxygen. And without the presence of oxygen, you're really limited in terms of how much breaking down of those carbon bonds from the glucose you can really do, right? Remember, we use that example of when you burn sugar in the presence of oxygen, so that there's oxygen with its greedy electron accepting tendencies to happily engage those electrons that are freed up when you burn glucose and you break the carbon-carbon bonds. If you have oxygen present, boy, those oxygens can happily take those electrons, right? But without oxygen, you cannot get all the energy out of glucose. And that's why we said the best that early life could do on Earth was get two crummy ATPs out of a molecule of glucose, right? And what you had left then were sort of like the two halves of the glucose molecule that you started with. And we learned that that is pyruvate, right? And life evolved to at least do something else with the pyruvate, right? You don't just throw it away. You can at least use the pyruvate as a dumping place to dump the electrons that you had taken off of the glucose intermediate molecules during glycolysis, right? Thereby freeing up these electron carrying molecules to go back and keep the glycolysis going. Right? So what we're going to do today is now discuss what can you do if, in fact, you have plenty of oxygen to work with, right? So not anaerobic respiration, but aerobic respiration, and how this involves a complicated set of pathways, including one known as the citric acid cycle. You guys have probably learned about the citric acid cycle before. It's also the Krebs cycle. You're probably introduced to it maybe in high school biology, certainly in biology like 171, but you probably didn't go into a lot of depth. You were probably just told that you have this thing called the citric acid cycle. So we're going to take a look at what that is today and why it's so important. All right, so remember, the absence of available O2 limits how much oxidation of glucose you can do, right? So cells that are in anaerobic conditions or hypoxic conditions, right? That's another way of saying low oxygen conditions, have to depend either heavily or solely on glycolysis. So you get a net gain of 2 molecules of ATP per glucose molecule, right, that goes through the process of glycolysis. But this is, like I said, pretty crummy because those phosphate bonds on those 2 ATPs that you've made from the glycolysis of 1 molecule of glucose, that energy that's now carried off as ATP, that only represents something like 3% of the actual potential energy that's in a molecule of glucose, right? Because by and large, those carbon-carbon bonds are still intact at the end of glycolysis. So let's review. On the left here, this is coming from glycolysis, and you have the end product of pyruvate, 2 molecules of pyruvate, right? Each one representing 3 carbons that came from the original 6-carbon glucose, right? That went into glycolysis at the beginning. Right? We said that anaerobic microbes evolved fermentation to at least get something out of the pyruvate rather than just stopping there. You cannot get more energy out of the pyruvate without oxygen to help you out, right? So you're not going to get more ATPs out of pyruvate through glycolysis, but at least you can go through fermentation, right? And we said that these NADH baskets, right? I describe them as these coenzyme baskets. These are these dinucleotide molecules that have a nicotinamide base on them. So they're nicotinamide adenine dinucleotides, right? And they carry pairs of electrons that were taken off of the glucose by enzymes during glycolysis. And you have these filled NADH baskets, right, that need to somehow be emptied if you're going to send these baskets back to help keep the glycolysis going. So at least the pyruvate is a convenient dumping place for those electrons. So we learned that cells have enzymes such as the lactate dehydrogenase or alcohol dehydrogenase that can facilitate the dumping of the electrons out of these baskets, turning the pyruvate into things like lactate or ethanol, right? And so fermentation, pretty much the sole purpose of fermentation is just to free up these electron carrying baskets so that the NAD plus can now go back and help out with keeping glycolysis going to get filled with electrons again. We learned that ethanol is sort of this end waste product of microbes, right? I conveniently call it like the poop of the microbes, because it is, right? Even the microbes don't even want the ethanol anymore because it's poison, right? It's basically like something that they need to get rid of, right? But, but whatever, we drink it, okay? In our muscle cells, we learned how if you're working your muscle cells too hard and they're burning up too much ATP or using up too much ATP, and there's not enough oxygen to help with aerobic respiration to kind of keep that supply of ATP up, we learned that our muscle cells can begin to lean more heavily on lactate fermentation, right? Because at that point, your cells still need more ATPs, but if there's not enough oxygen, right, getting to your leg muscles or your arm muscles because they're so strenuously working out, right, then it's got to lean more heavily on glycolysis, right? So through channeling pyruvate into lactate fermentation, your muscles are doing the same thing that microbes do. They free up these electron carrying baskets so that the NAD plus can go back and at least make sure that your glycolysis is going at maximum efficiency in those muscles. So again, this is what you're limited to if you don't have any oxygen to play with. Oh, but we said right? Over the first billion years of life on Earth, when photosynthetic organisms were taking energy from the sun and then CO2 and H2O and making glucose molecules as their raw fuel source, right? What's left over when you take 6 molecules of CO2, 6 molecules of H2O, and arrange them to make a molecule of glucose, which is C6H12O6, you have 6 O2s left over, right? And so that is also a waste product as far as the photosynthetic organism is concerned, right? It just needs to get rid of it. Right? So over the first billion years of life, the atmosphere of Earth began to fill up with more and more O2. So suddenly you had options, right? The presence of O2 allows for additional opportunities to get more mileage out of that original glucose molecule that you had up until this point stopped at just having 2 molecules of pyruvate. Right? So aerobic conditions, aerobic respiration, we as organisms that depend upon aerobic respiration, we are called obligate aerobic respirators, right? Meaning that many organisms, you have to have oxygen in order to be able to make enough ATP, right? Just being stuck with glycolysis alone doesn't work to maintain our bodies. Right? So if you have oxygen present, then you can start off where the glycolysis left, right? So the glycolysis ended with pyruvate. Instead of sending pyruvate through fermentation, if you have oxygen available, you can do something with these 3 carbons here, right? The carbon-carbon bonds that are in there. You can bust those up, right? And get more energy out of them. And so the way that works is our cells take the pyruvate from glycolysis and it converts that pyruvate into an intermediate molecule known as acetyl Coa, right? So you can see the acetyl Coa has 2 carbons out of the 3 carbons that were in the pyruvate. What happened to the 3rd carbon? This one up here. Well, notice, what is the carbon bound to? It is a carbon bound to 2 oxygens, right? So if you cut this top carbon off, what do you have? You have CO2, and that CO2 is then released, right? In that process, you've now freed up 2 more electrons, right? By breaking this bond, and those electrons are carried off by an NADH plus empty basket that carries them off as a filled NADH basket, all right? So you can see you're beginning to tap into more of that energy. This acetyl CoA then feeds into what we call the citric acid cycle. And that's where the oxygen becomes super important. Right. Really quickly, because like I said, there's a lot of stuff to go over here. I think many of you might be surprised to know, right, that all of the cells in our body still begin with this ancient glycolysis, right, as the first steps in breaking down the glucose. You might have just assumed, hey, why are we learning about this glycolysis? Shen makes this big deal about how ancient microbes invented glycolysis because there's no oxygen, right? Why is that even relevant to us now? Who cares? It's relevant to us because every single cell in your body still begins with glycolysis and gets 2 ATPs out of the glucose, right? Ends up with pyruvate as the end of glycolysis, and now sends that pyruvate into aerobic respiration, right? And you might think, why is it that we still have to start with glycolysis? If we have all this oxygen now, why didn't life just switch over entirely to aerobic respiration? The simplest way of thinking about it is because evolution of life doesn't work that way, right? Never could you imagine that at some point in evolutionary history of life, just because there's enough oxygen, that suddenly all the cells could somehow magically switch over and come up with a new way of making ATPs directly through aerobic respiration, right? Because those cells still needed to survive. An analogy is, for example, automobiles, right? The steering wheel and the gas pedal and the brake pedal, right? We don't even second guess a steering wheel and a brake pedal and a gas pedal. We're so used to it, right? Because nearly all cars that we drive, That's what we're used to. Cars have been around in their modern form since maybe like the late 1800s, early 1900s. And around that time, the limitations of technology were such that, you know, the best you could do is have a steering wheel, right? And separate pedals for your feet, right? But now, like whatever it is, 200 years later, right? Or yeah, something like that, we certainly have way more advanced ways of controlling things, right? I mean, just think about your tablets that you can swipe things with your finger. I mean, those of you who like to play video games, think about the fact that even like a video game controller, you're doing so much just with your thumbs, right? Having a steering wheel, gas pedal, and brake pedal, and having to kind of juggle, you know, moving your hands and your feet, if you think about it, that's not very efficient, right? And yet, all cars for generations have been based upon that, because once you start down a particular path of using a particular mechanism that is so fundamental to the operation of, let's say, the car, it's very hard to scrap that entirely and start with something new just because you now have the technology to do something new, right? Just consider if next year all the auto manufacturers like, you know, Toyota, Honda, Ford, GM, whatever, suddenly said, all the cars we're going to make from now on, the new models, don't have a steering wheel, don't have a gas and brake pedal anymore. It's all touchscreen. What would happen, right? I don't know about you, but I wouldn't be very comfortable making that jump, right? And that's why those types of mechanisms in cars have stuck around for so long. And that's why life on Earth, for example, our cells are still stuck with doing glycolysis first, but evolution builds upon that, right? Rather than erasing it and starting from scratch. So aerobic respiration builds upon the pyruvate that comes out of glycolysis. Woo! I said I needed to give a quick example, but then I think I took up a lot of time. All right. So I said glycolysis yields pyruvate, right? So here we have a molecule of glucose goes through glycolysis. You get two crummy molecules of ATP. You also end up filling one of these NADH baskets or NAD plus baskets with the two electrons that come off. You end up with pyruvate, right? If our cells have plenty of oxygen available, right? As we're sitting here, me lecturing, you listening to this, right? You know, hopefully you're not experiencing labored breathing, or you got plenty of oxygen, right? Your cells will then take that pyruvate and send it down this pathway of making acetyl CoA. So the acetyl CoA is two of those carbons from the pyruvate, right? The third one popped off as CO2, right? And you see it's attached to this sulfur group and this CoA. What is that? This stands for coenzyme A, right? You're like, coenzymes, we've talked about that. Coenzymes are small molecules that play an important role in helping enzymes do their function so that a pathway of different biochemical reactions can be chained together efficiently. All right. So let's sort of summarize this part. What we're saying is that the way our cells utilize glucose in our bodies, right, starts with glycolysis, that primitive anaerobic way of getting some ATPs out of glucose without the need of having oxygen. So you get a very modest yield of ATP, right? 2 ATPs per glucose, and you end up filling these NAD plus baskets, and now you have these filled electron carriers that are floating away, right? So through the 3 phases or stages of glycolysis, you end up with pyruvate. Now, because you have lots of oxygen available, right, we evolved these next steps. So step 2 is the creation of acetyl CoA from that pyruvate. 2 of the carbons from the pyruvate attached to this coenzyme A molecule that we'll look at the next slide, right? That acetyl CoA gets fed into the 3rd step here, which is the citric acid cycle, right? And the citric acid cycle is going to involve the carbon-carbon bonds from the acetyl CoA, right, being manipulated and being put on different sort of locations on a series of intermediate carbon molecules. And in doing so, you are going to generate many electrons. Why? Because remember, when you have electrons involved in a bond on a complicated molecule, if you have, let's say, a carbon that's stuck at a particular spot on a molecule, and that bond of the carbon with whatever other carbon is part of the main scaffold in that molecule, right? The energy of that carbon-carbon bond may differ depending on where on that big complicated molecule you're sticking that carbon, right? Because a carbon-carbon bond alone, right, might have a particular value of energy if it's literally just two carbons floating around. You're talking about the energy. But if one of those carbons is attached to a big bulky molecule filled with carbons, you're going to have all kinds of factors that are actually going to contribute to the energy of those electrons in that carbon, right? So if you start moving that carbon to different places on complex molecules, you end up changing the amount of potential energy available, right? So then if you break those bonds at certain times, you can tap into that energy, and the electrons that are taken off go to fill more of these coenzyme carriers like the NAD pluses, right? Those electrons then are going into step 4 of aerobic respiration, and that is the electron transport chain. Right? Those filled electron carrying baskets are going to get dumped out, right? Dumped out because eventually you have oxygen, right? You're breathing in oxygen. Oxygen is there as one of the most greedy electron accepting atoms, right? Only fluorine, F, is more stronger than oxygen in terms of wanting to take electrons and being very greedy and hanging on to them. Right? So you can dump out these baskets and give those electrons to oxygen, but the key here is you're not going to do it directly, right? You're going to make these electrons flow through a series of big membrane protein complexes that are serving as proton pumps. So as the electrons flow through this series of proteins in the membrane, they're going to make these proteins pump protons across, building up a very strong proton gradient. And then step 5 is going to be that proton gradient is going to be allowed to flow back downhill through the membrane, providing a very strong force, right, that can then be used to power other proteins that actually make ATP in very high throughput amounts, right? So that's kind of the overview, right, to kind of ease you into this. This is what we're going to talk about for the rest of today and on Monday. Right? So let's compare 2 different strategies for making ATP. Glycolysis makes ATP, those 2 crummy molecules of ATP, without the help of oxygen. How does it do it? It does it because you have enzymes that participate in glycolysis that quite literally take 2 different substrates to work on. Right? One of them would be one of the intermediate molecules in glycolysis, for example, 1,3-bisphosphoglycerate, right? And you're like, that sounds familiar. Yeah, if you go back to the glycolysis pathway, you'll find 1,3-bisphosphoglycerate as one of the intermediate molecules, right? You'll have this big purple enzyme here that can bind 1,3-bisphosphoglycerate. At the same time, it binds another substrate, adenosine diphosphate, ADP, that has 2 phosphates on it, right? And this enzyme then will catalyze, right? It'll speed up the transfer of one of these phosphates off of the 1,3-bisphosphoglycerate, right, turning it into just phosphoglycerate, right? And importantly, that phosphate group is going to get added to the ADP to now make ATP, right? So this is what we call substrate level phosphorylation, right? The enzyme is working on these as its substrates, right? And it's almost like you're hand making each ATP, right? Imagine that the enzyme is like hand making each one from grabbing a 1,3-bisphosphoglycerate and grabbing an ADP, right? So the pros of this, hey, it works, right? You've made an ATP molecule, but the cons are this is not very efficient, right? Because think about it, at any given moment, how many hundreds of thousands of enzymes inside of the cell are just waiting to get an ATP molecule for them to do their job, right? And if you have this enzyme in glycolysis just taking its time hand making each individual ATP, that's not very good. That wouldn't be enough, right, to power very complex functions. Right? What if you had some sort of high throughput way of making ATPs. So imagine these turnstiles, right? Like you'd find in a subway station or maybe entering some sort of sports arena, right? And let's say you have a big crowd of people just waiting to get in, and they all have to flow through these turnstiles, turning the turnstile, right? If you could hook these turnstiles up to generate or do work, boy, you could have a really high throughput, efficient way, right, of harnessing energy, right? And so that's what we do in aerobic respiration. We have these proteins here that are sort of in this light gray in the picture that are part of what I just introduced on the previous slide, the electron transport chain, right? You're going to have electrons that are brought to this chain by NADH baskets that are filled with the electrons, dumping those electrons into this chain and through a whole series of proteins. This cartoon only shows one of them here, right? But we'll study that there's actually a series of these proteins that basically pass the electron to each other. And as the electrons flow through them, that's what gives them the power to pump protons against the gradient, right? Building up a proton concentration on one side of the membrane that is like 100,000 times stronger than the proton concentration on the other. And just like the concept of indirect active transport that we learned about previously, right, you will have other protein complexes like this purple one here, right, that will allow those protons now, right, to very eagerly flow back downhill through the membrane from high to low. And these are the turnstiles, because as the protons flow through them, these turnstiles will spin, and suddenly you can make a lot of ATP by putting phosphates onto ADPs, right, in a very sort of like mass production fashion, right? So if these sound really sort of like, whoa, that's intense, I don't quite get that, don't worry. This is just an introduction to it. We're going to look at these in more detail, right? More nauseating detail. Okay. So where is this taking place? Right? Aerobic respiration takes place in the mitochondria of your cells. Look at this illustration on the left. So here's a typical cell from the human body. Glucose comes in from the outside, and remember, it's maybe transported across the membrane through something like a carrier protein like GLUT1, right? So it's moved to the inside. In the cytoplasm, this is where glycolysis takes place. All right, so the three phases of glycolysis that go to work on this one molecule of glucose takes place in the cytoplasm, and you end up with pyruvate, right? The end product of glycolysis. That pyruvate is then moved into the mitochondria, where all the rest of this good stuff is going to take place. The mitochondria, shown here, actually has two series of membranes, an outer membrane and also an inner membrane, and that divides it into sort of two spaces, right? Sort of a intermembrane, an inner space, and also an outer space, right? And so between the two, you can think of this as an intermembrane surface, okay? Studded on here, you see these little dots, right? These are ATPase pumps, okay? And these are the what we call F-type ATPases, right? So in the illustration over here, you are going to have these pumps pumping protons from the inner membrane space through this intermembrane here to the outer membrane space, building up that super strong proton gradient, right? Then you will have other proteins that will now allow those protons to happily flow back downhill through the membrane. And in doing so, it's coupling that action to the creation of ATP, right? And this happens in our mitochondria. Right? I love this illustration. There's very few illustrations from the book that I think that they actually did right. You know, most of them I'm like, I would have done it a different way. But this one I like a lot, okay? And I know it looks intimidating at first, right? But I think this illustration will become your friend because it is a great illustration to help you review this whole process of aerobic respiration. So what we have here is a zoomed in section of a mitochondria, right? So this black square is what we're looking at blown up on this big image. So the light sort of tan outside would be the cytoplasm of the cell, right? So glucose has been brought into the cytoplasm of the cell. That's where glycolysis takes place, right? Step one of the 5 steps of aerobic respiration starts anaerobically with glycolysis. And so you make a couple of ATPs, right? And importantly, right, you fill up a couple of these NAD baskets to make NADHs, right? That happens in the cytoplasm. These baskets can now flow into the mitochondria along with the pyruvate. That pyruvate is moved into the mitochondria, right? And it is converted into acetyl CoA. And in the process, right, you're going to fill more of these NADH baskets with electrons. You're going to release a molecule of carbon dioxide, and you've made acetyl CoA, which now feeds into step 3, which is the citric acid cycle. Okay. During the citric acid cycle, you start breaking some of these carbon-carbon bonds that are on these big intermediate molecules that are there as part of the citric acid cycle, right? And you're generating more carbon dioxide off of it. You also manage to generate 1 molecule of ATP for every sort of circle or cycle of the citric acid cycle. So clearly, the citric acid cycle is not about generating tons of ATP itself, because in fact, where's the oxygen here? There is no oxygen involved yet, right, as part of citric acid cycle. What you're really doing is you're filling up a ton of these electron-carrying baskets from the electrons that you've taken off of these big carbon intermediate molecules as you are breaking and moving carbon-carbon bonds around, right? Those filled electron baskets, these NADHs, and there's another type of coenzyme called FADH, right, FADH2, all of these baskets then go to the electron transport chain, right, which involves these tan-colored proteins that are in this intermembrane, on this intermembrane that separates the outer or the outer from the inner mitochondrial space, right? These electrons then flow through this chain of proteins, and as they flow through, these proteins use the electrons as power to pump protons, right, across the membrane. So you end up building up a ton of protons in the intermembrane space here, right? Then you will have other proteins, right, that are designed to now allow those protons to flow back downhill. Right? And in doing so, as they flow downhill, these are like all the people moving through the turnstiles, right? They are designed to now crank out a ton of ATP, way more ATP than just the lowly 2 ATPs at the start that you got from glycolysis, right? All right, so let's take a look at the citric acid cycle today, and then next time we'll look at more depth about things like the electron transport chain and ATP synthesis. So the citric acid cycle, what's going on here in step 3? All right. So it all starts with pyruvate from glycolysis being converted into acetyl CoA, right? And the acetyl CoA is what's fed into the citric acid cycle. All right. I know this looks scary, but it's actually not. Look, here is pyruvate on the left, right? 3 of the carbons that came from the original 6-carbon glucose. Remember, at the end of glycolysis, you end up with 2 3-carbon pyruvates. Essentially, the glucose has been split in half, right? And you have these 2 pyruvates. Look at the structure. Part of the pyruvate, well, it's made of 3 carbons, right? Part of them, this bottom 2, is an acetyl group, right? Because it's a carbon with a methyl group down here, double bound to an oxygen. The other one is a carbon bound to 2 oxygens. So right off the bat, right, I already mentioned this is carbon dioxide, right? So if you go ahead and cut this bond off, right, you liberate or release a molecule of CO2, and then you're left with this acetyl group, right? This big thing up here in the purple outline is coenzyme A, right? And look, some familiar features. This looks like some sort of nitrogenous base. There's a ribose-looking sugar type of thing, has phosphates attached to it. Again, a wonderful reminder, right, that when Mother Nature stumbles upon certain molecules that are useful, like attaching a nitrogenous base to a sugar and putting phosphates groups on it, you can make nucleotides with it, right? And you can also make this coenzyme A with it. Of course, there's this big long chain of carbons attached to the end of it, and that's what makes it coenzyme A. This is where the acetyl group over here from the pyruvate, right, after it's been cut so that the carbon dioxide part of the pyruvate is booted out, right, this acetyl group that's left gets added to the coenzyme A, right? And so in this process, you're going to fill another one of these NAD electron baskets representing the electrons that were sort of cut, right, when you release the CO2 over here. And this acetyl CoA now that consists of 2 carbons, right, which are the acetyl group, attached to this big coenzyme A on the top, right, now gets fed into the citric acid cycle. Importantly, what we're dealing with here are 2 carbons attached to coenzyme A, right? These 2 carbons don't have to come from a sugar. Right? Other metabolic molecules can also be converted into acetyl CoA. For example, proteins can be broken down to make acetyl CoA. Fats, right? So when you talk about burning fat, right, what are you talking about? You're talking about converting big triglycerides, right, big fatty acids, into small little double carbons that can then be made into acetyl CoA. In other words, the burning of fat simply means you are taking the energy that was stored in that fat molecule and you're ready to use it to make ATP, right? Because it's already these carbon-carbon bonds, right, in the fat, you don't have to go through glycolysis because glycolysis is meant to work on sugars, right? You can feed these carbon-carbon bonds from fats right into the citric acid cycle by turning them into acetyl CoA, right? And what's this? This is a ketone. Remember, for organic chemistry, oh boy, bad flashback time, organic chemistry. A ketone is, the basic structure would be 3 carbons, you have a double bond, right? R just means you could be any sort of group that has carbons on there, right? The simplest type of ketone would be if these 2 R groups are methyl groups or CH3s, right? And that would be acetone. Right? So that's why you look over here, right? That's essentially why this is called an acetyl group. So acetone would be if you have 2 CH3s over here. What's acetone good for? Well, it's a good solvent. So those of you who use a nail polish remover, right? Obviously water can't be used to remove your old nail polish, right? Because the nail polish, typically the formulas are these big, long hydrophobic molecules, right? So water won't really do anything. But if you use an organic solvent like acetone, right? Acetone is polar because it has this greedy oxygen on one side. So this side would actually play very well with water molecules. Whereas the other side, if they are methyl groups, right, are hydrophobic. So you would end up having your acetone in your nail polish remover, right, coating all of the long hydrophobic molecules in the nail polish, in your dried nail polish, right? And suddenly now, if you put water on top of it, right, the water will happily bind to the acetone that itself on the other side of it is bound to your nail polish remover, nail polish. And that's how the nail polish then becomes liquefied, right? It becomes soluble in the water because it's coated with that. Ketones, of course, some of you might not have automatically thought of nail polish remover. Some of you might have thought of, ketone diet, right? Or keto diet, right? What's that about? That has to do with the fact that your body is designed to be able to take ketones like this here, right, and convert them into acetyl CoA to power ATP production through the citric acid cycle without bothering with having to mess with metabolizing sugar, right? So keto diets that are very heavy in fats, like 70% fat or something like that, right? A lot of it is these fats are being broken down into ketones, right? And these ketones then can be converted into acetyl CoA, right? And that feeds into the citric acid cycle. All right. That was a little extra bonus stuff there. Oh boy. Okay, I didn't even prepare you for this, right? So here is the citric acid cycle in all of its nightmarish glory, right? So don't worry, you don't have to memorize this. You're going to have to memorize certain parts of it in a simplified form, and I'll show you that simplified form at the end. But I want to show you the big, scary, detailed one first, because this is how you can see where a lot of cool stuff is happening, all right? So the citric acid cycle, you can see here, has a number of enzymes that participate, right? And they're just called, and this one here, citric acid cycle enzyme 1, 2, 3, 4. You go all the way around to citric acid cycle enzyme 8, right? And this is the name of them in the middle. You don't have to know the names of these enzymes, but you do need to know the names of these intermediate molecules, right? Citrate, isocitrate, alpha-ketoglutarate, succinyl CoA, succinate, fumarate, malate, and oxaloacetate. All right. Let's see what's going on here. So up at the top, we have the pyruvate that came from glycolysis, right? So it represents 3 carbons, half of the glucose, and you're going to have 2 pyruvates that were made from glycolysis, but we're just going to follow 1 in this illustration, right? We said that one of the first steps of converting pyruvate to acetyl CoA is you boot off that carbon dioxide. So you've lost one of those carbons, right? And in the process, you filled an electron carrying basket with the energy that was liberated by breaking that carbon off, right? Then you're left with a 2-carbon molecule, which is basically an acetyl group that gets added on to CoA. And this acetyl CoA now gets fed in. I like the fact that they go ahead and color code certain things, right? Anywhere you see the formation of a carbon dioxide, they've conveniently put that in dark gray, right? So you can see that this carbon dioxide up here came from the carbon and the 2 oxygens that were booted off of the pyruvate, right? In pink are the 2 carbons from the pyruvate that you're left over that are actually being fed into the citric acid cycle as part of acetyl CoA. And throughout this cycle, they keep those 2 carbons highlighted in pink to help you sort of visualize what's going on, right? So you have this big molecule waiting for it that's called oxaloacetate, right? You can see that it is a 1, 2, 3, 4 carbon molecule, right? The 2 carbons from the acetyl CoA, those 2 pink ones, get added onto it, and you end up with citrate, okay? Then the next enzyme begins to move some of these bonds around, right? Sort of playing with the energy of these bonds. You can see it gets rearranged into isocitrate, right? So iso, when you say something is iso means what? Same, right? Or identical, right? So this is citrate, but a different arrangement of the bond. So it's isocitrate. Then look what happens. You actually get rid of the first carbon dioxide that's off of it here. So this carbon dioxide gets booted off, right? And the energy that's liberated by breaking that carbon-carbon bond here goes to filling an NAD basket, right? So those 2 electrons that were participating in holding this CO2 onto the scaffold now went into an NADH basket, right? Then you end up with alpha-ketoglutarate, okay? And you go ahead and have another enzyme that breaks this other CO2 off. So that other CO2 is liberated, right? And those electrons, again, are carried off in an NADH basket. Right? That leads you to the succinyl CoA. So this now binds to another coenzyme A molecule, right? So that's why it's succinyl CoA. Right? And then that CoA gets broken off by this other enzyme that is able to use that breaking of the bond to actually make one ATP molecule from an ADP that was floating by, right? It grabbed it and it made an ATP, again, through phosphate level phosphorylation, right? Just like the way the enzymes did a hands-on, handmade approach of transferring phosphate groups, like in glycolysis. Right. Now that the succinyl CoA has lost the coenzyme A part, right, that's why this little arrow shows that the coenzyme A goes back this way, you are left with succinate. Right? Notice at this point, you still have the 2 pink carbons that came in from the very top. These are the 2 pink carbons that you can think of as coming from the pyruvate that originally came from the glucose, right? And you might find it strange that up until this point, those 2 carbons are still essentially intact, right? But now what happens is you begin to rearrange the bonds so that technically now the arrangement of those bonds is not the same as what those 2 carbons started at. Again, because you're beginning to play with changing the energies of these bonds, right? So you get to malate, right? And look at what has happened through the rearrangement of these bonds, right? You've now basically restored 2 carbon dioxides, or you're getting to the point of restoring 2 carbon dioxides on this molecule, right? And then the last step is you convert the malate into oxaloacetate, where you started from, right? And now you have 2 carbon dioxides on there again, right? So you might look at this and you'd be like, wait a minute, this is like anticlimactic, right? Because I thought the whole point of fully oxidizing glucose was we were going to blow apart all the carbon-carbon bonds, right? That's my mistake for making you think it's like you're burning a marshmallow, right? And it's this big, violent kind of reaction. But remember, I taught you, right? In our cells, you can't have violent, super high exothermic reactions like that, because how are you going to harness the energy of that, right? Everything is done step by step by step, working on individual bonds, oftentimes just moving those bonds around to play around with the energy of those bonds until the energy of those bonds are sort of just right, that you then break the bonds, and those electrons that were at that energy state in that bond right now can be moved onto another molecule and carried off to power something else. Right? But you say, but still, I don't get it, because it's weird how these two carbons that came in, right, from the glucose molecule, right, ends up just sort of being like absorbed into these intermediate citric acid cycle compounds, right? And that's true, right? So that's why I say it's kind of anticlimactic, because when you look at this, you don't really see how is this actually breaking up all the bonds of that original glucose, right? Well, think about it this way. I use this analogy. Imagine you have a market, a supermarket, grocery store, right? And you have some shelves that are lined with soup cans, right? And let's say in the morning, the manager or the owner of the market, this guy over here, he goes into the back, right? And he has in his inventory pallets of Campbell soup cans. So he'll go ahead and take soup cans and he'll stock them on the shelf, right? And he'll fill up the shelf so it looks nice and packed up, right? Throughout the day, customers come along and they will take maybe 1-2 soup cans off, right? Buy them and leave the store with them, okay? The next day, the manager, same thing. If there's empty spaces on the shelf, he'll just replace them with additional soup cans, right? That means at the start of the week, if you came in Monday morning and looked at the shelf and saw that it was all filled with soup cans, and then you came in at the end of the week, Friday afternoon, and looked at the shelf, and you're like, wait a minute, it's still filled with soup cans, right? What happened, right? That's not using up the soup cans, right? So imagine the soup cans are the carbons that enter the citric acid cycle as the 2 carbons in acetyl CoA, and these now end up sort of being absorbed in a way or placed, right, on different positions within the different intermediate molecules of the citric acid cycle. So as the citric acid cycle goes around, right, carbons are being taken off at different points, right? And importantly, new carbons are coming in from the top all the time, right? Just as long as you have more soup cans, or I should say, more acetyl CoA that's coming from your cells constantly using sugar, right? So think about it that way. Eventually, right, these soup cans are used up. Right? How would you know that? Let's say the manager ran out of soup cans on Monday morning, right? And through the whole week, right, customers were just taking all the soup cans. You know, it's kind of like if there's a big run on soup or something like that, you go to the store and you're like, oh, the shelf is empty. There's like 3 soup cans left. One is dented, the other one's sadly tipped over, right? So I joke about that, but what would that imply in your living cells, right? If you stop replenishing the carbons going into the citric acid cycle because you no longer have carbons coming in as a fuel source, either through glucose or through other intermediates like fats, ketones, proteins, right? The citric acid cycle would grind to a halt. And as a result, you would not be making electrons to power the production of ATP. And as a result, without ATPs, what happens to all the proteins and enzymes in your cells that rely upon ATP? They can't work. The cell dies, right? And if enough cells dies, your tissue dies, your organ dies, and you die, right? That's why we need a constant source of carbons from our food. Okay. I like the soup can analogy. Hopefully it works for you. Okay. So this is the last slide for today. It's kind of miraculous that I actually made it on time, right? So this is the simplified version of the citric acid cycle that we here in Bio 275, right, are expected to know. Okay? So the way I've drawn it is that I've labeled the intermediate compounds or the intermediate molecules that are part of the citric acid cycle. I've put in parentheses little reminders of how many carbons make up those. So I need you to know that. I need you to know that citric acid fits in on the citric acid cycle at this position here, and it has 6 carbons, right? I've also shown you that here's the acetyl CoA coming in, right? That's going to join up with oxaloacetate, right, to form that citric acid, right? 4 carbons from here plus the 2 carbons from here gives you 6. I also show you where those electron carrying baskets are being filled along the way, right? By NADH baskets carrying away electrons, and then this other FADH2, which we'll talk about next time, that also carries off electrons, right? And this is a reminder that in the process, there is one step where enzymes can do substrate level phosphorylation to just hand make 1 molecule of ATP with every turn of the cycle, right? So this is what you need to know. And remember that there are actually 2 acetyl CoAs coming into 2 different sort of instances of the citric acid cycle, if you want to think about it that way. Maybe that's actually not a good way of thinking about it, because after all, it's not really like a physical chain that goes around and around, right? It's just all these molecules are all mixed up in there, and these molecules and enzymes are all mixed together. And if an enzyme happens to meet up with the particular substrate or intermediate molecule that it recognizes, it does the next step, right? So in a living cell, you would actually find that all these steps are happening at the same time, you know, based upon how many instances of different intermediate molecules you have, right? But from 1 glucose, you get 2 pyruvates. Those two pyruvates then mean you make 2 acetyl CoAs, and then those 2 acetyl CoAs enter the citric acid cycle and then go through this, right? Each one going through what you see on the cartoon here.